We are going to have a discussion about automation and immunodiagnosis in microbiology. If you're taking a technician exam, you really won't have a lot of this information on your exam. If you are taking a technologist generalist exam for chemistry or chemistry exam or serology exam, some of this information will be more important to you. You know how I do it. I teach to all the levels at one time. It's easiest for me. You just have to pull out what you don't need. So again, technician, you can kind of gloss through this, just kind of know the high levels of it. And if you're taking a technologist, you need to know a little bit more. What everyone needs to know is this part about demonstrating knowledge of the API system and automated systems, because that's just a general thing that everybody has to kind of know. How do you identify microorganisms outside of biochemical tests that are done manually? The biggest test system used is the multi-test system. The multi-test system is a combination of both manual and semi-automated procedures. In some of them, you inoculate a strip and then you incubate it, and then you interpret what the results are. And for some others, you inoculate it and you get a reading right away. The multi-test system is really important because it's considered to have a shorter incubation time and reaction time. That is by comparison to growing bacteria out, waiting 3 or 4 days for a good sample and then inoculating. It's basically compared to the old method of doing things. So the multi-test system uses biochemical and enzymatic reactions. For those of you who, like I said, doing chemistry, you're going to have to know a lot of the enzymatic reactions, so you should get used to that now. And this is more reproducible and consistent results because there's not the person making the inoculation and then reading and interpreting based on their thoughts. It is actually clear cut based on a guide that is given for what that system should read. So for instance, there's systems that are set up just for Enterobacteriaceae, and based on the color scheme, you would say that this particular organism is present. Example of multi-test system, the biggest one, bioMérieux. bioMérieux makes so much. So bioMérieux has an API strip, and it comes with microtubes with dehydrated substrates that are reconstituted by adding a bacteria suspension, which means you have to take the bacteria and place it in something like saline, and then you place a portion of that sample into this strip. There are systems for individual groups of bacteria, such as Enterobacteriaceae. There's an entire system for non-fermenters, an entire system for Neisseria, Haemophilus staph, strep, Corynebacteria, anaerobes, yeast. So again, these systems exist for different organisms, but you have to know which organism you're looking for. You're going to know that based on the sample that was submitted, the diagnosis that was submitted, and then therefore you know which strip to actually inoculate. And of course, this is going to come after you've already done your gram stain, and you're going to see that you have like gram positive, gram negative, cocci bacilli, yeast present, and then you're going to actually have organisms that grow aerobically or anaerobically, and then you're going to know more specifically which one of these you should utilize. The good thing about this system is that there's a database that is web-based, so you can always find answers in the middle of the night. You're not really sure how to interpret it, you can go online and find answers to help you with interpreting the system. Here is what an API identification strip looks like. Notice there's different colors. Just like we briefly discussed fermentation being yellow and pink and colors like that, you can also see fermentation and those color wells in certain API strips. Some of these are indicators of fermentations, or some of them are just because of the reagent that's utilized and it turns a different color. So it's very, very helpful. There's also what's called BD-BBL crystal system. Now, do you have to know every system and every name? No, you have to know what it does. So this one has biochemical tests and a tube, and it's what's called a one-step inoculation. So you just inoculate one time and it runs all of these tests. But here's the big thing is that it can identify over 500 organisms manually or by using a auto reader, which most microbiology laboratories have an auto reader that we put it in. So it's used for gram negatives, enterics, non-fermenters, and gram positive aerobic organisms. Here's what the BDL crystal looks like. See, it's kind of similar, but not exactly, but there's color changes going on here. Most huge laboratories are going to use automated systems, and this is going to help you when you actually go and work into the laboratory. So automated systems identify antibiotic susceptibility as well as identifying the bacteria. Antibiotic susceptibility means, okay, if I were to treat this patient with this particular antibiotic, how effective would the antibiotic be on the organism? So with some automated systems, you can find that information out at the same time that you're finding out what organism is present. The principles utilized are colorimetry, nephemetry, and fluorimetry. For those of you who are taking serology, you are going through the process of learning about these, but for those of you who have not taken serology, of course, you will learn a lot more about these methods. We will briefly touch on them in micro and chemistry as well. The microscan is one of the most popular automated systems. Of course, it's made by Siemens, which right now they corner the market in almost everything laboratory. There's another one, Trek Diagnostic has a system, BD Phoenix. So this is just giving you some names. Again, microscan is the one that's important because a lot of people have it, and then Phoenix, a lot of people have that too. You don't have to remember all of them, but just kind of be aware that they exist. What you have to know is when we get into the details of the principles, nephelometry, what it is, and that they can provide susceptibility testing. Vitek, oh God, Vitek, I've used Vitek for many, many, many years. Very useful. Vitek is good because most of your clinically significant bacteria and yeast are identified with the Vitek system, and you can pretty much walk away from it. It's an automated system. It grows it out. It has different reagent cards for identifying the organism. And of course, the principle, nephelometry. There's also the microscan walkaway by Siemens. It's fully automated, uses colorimetric and fluorimetric principles, which, so again, this is what you're going to need to know. These systems, what that actually means, how it works. So the basic things are automated systems. The principles are colorimetry, spectrophotometer, measures color change, and pH indicator. So the pH indicator causes the color change. So going from yellow to say orange, or going from um, no color to blue, or going to a pink color. And sometimes there's the presence of a substrate or enzyme is used. Then there's other systems, nephelometry, light scattering, and turbidity measurements. So with fluorometry, substrates have a fluorescent component that is used to detect an enzyme, and if the enzyme metabolizes into the substrate, so it gives a color change. Nephelometry is light scattering, and it measures turbidity, meaning how thick the light is. So if there's a lot of growth there, you won't be able to get a lot of light through the growth. But if there's not as much growth, then more light comes through. In general, immunochemical methods involve immunology, which is antigen antibody reactions, and chemistry. So there's some type of chemical substrate, enzyme, or something that is added in order to make the reaction take place. So basically for micro, you use an antigen and antibody reaction to detect the microorganism when the infectious agent cannot be cultured on artificial media. So if there's something that is entirely, one, too dangerous to grow in the main laboratory, or two, we just cannot provide the optimal temperatures for it to survive in a laboratory, this is the method that works really well. The antigen normally respond. So here's, if you don't know, if you haven't taken some of the other courses, you don't know what an antigen is. Antigen is a foreign compound that stimulates an immune response, leading to the formation of antibodies. So what happens is that something enters your body that your body is not aware of, and then your body's like, what is this? And so your body is like, I want to get rid of whatever this is because it doesn't belong here. And it makes what's called antibodies wings against. So something to fight that thing that invaded its property, so your body's like trying to get rid of it. In microbiology, we use that as a means of identifying an organism that your body has come in contact with and made antibodies to reject it. Antibodies are proteins, and they are formed in response to stimulation by the foreign antigens. There's a concept that you will come to know a lot about, and this is not the only course where you have it. Affinity and avidity. When it comes to energy antibodies, those two terms are highly, highly, highly important. Affinity is the strength of the reaction. The strength of the reaction between a single antigenic determinant and a single combining site on an antibody. So it's a one-on-one reaction and how strong that reaction is. The end result being the forces of attraction and repulsion in the antigen-antibody reaction. Notice I said antigen-antibody reaction. You can also say antibody-antigen reaction. It just depends. The avidity is the strength of binding of many determinants, right, and antibodies. So it is the sum total of all binding between antibody and antigen. I'm going to go through that again. The affinity means how likely this particular antigen is going to combine with this one particular antibody. How strong of attraction do this one antigen and this antibody have with each other? Avidity is like your social group at school. So at school, you have a group of friends, and they like to characterize the mean girls, the cheerleaders, and things of that nature, the jocks. Which group do you most likely fit in? That's the case with avidity. The strength of binding of many determinants in antibodies. So it's the sum total. Okay? Hopefully those analogies help. Immunochemical methods involve an epitope. This is the part of the antibody that reacts with the antigen. Again, this will come up again. And it involves specificity. Specificity means specific, meaning the ability of the antibody to distinguish between antigens that have very small differences. So that says that although I have twin nephews, and my twin nephews look identical, my twin nephews wear their hair identical, my twin nephews walk identical, my twin nephews, most people cannot tell them apart, but I can tell them apart. There's one very specific characteristic that they have. and that is the way they greet me. I know specifically that one is not gel and one is not sear simply based on how they say hello. That's a very small difference between two identical human beings. And that's what immunochemical methods do. You need to be identify very, very tiny differences because most of the organisms are very similar. They have very similar biochemical reactions, and it gets down to one test that makes it not be the same organism as the first one. The problem with specificity is that antibodies with low specificity may be associated with false positive reactions. Of course, we want to eliminate those. There's another problem with immunochemical methods, and that is cross-reactivity. Cross-reactivity means the antibody is specific for one antigen, but it reacts with an antigen with a similar, non-related structure. Back to my twins. My twin nephews, they are identical. And if you saw them in the street and you saw them from a distance, you might run and hug Nigel thinking he's Nasir. But they're different. Cross-reactivity is something you have to worry about because the treatment for the different disorders is different. Antibody susceptibility determines which drug is used to treat the patient. But if you think it's the wrong organism, then you're going to mistreat the patient. Here's another term, sensitivity. Sensitivity is the ability to detect small amount of antigen or antibody. Test systems with high sensitivity are better able to detect small quantities of the substance being analyzed. So the sensitivity wants to know that, yes, there's Staph aureus here, even if there's like a tiny bit. Now here's the kicker. What does sensitivity and specificity in antigen detection mean? Sensitivity is the measure of the true positives because sensitivity measures a tiny, tiny, tiny amount of antigen present, right? So this, and this can be kind of confusing, but honestly, sensitivity and specificity is used every day in the laboratory, not just for micro. Definitely for microbiology, but for some chemical reactions as well. So TP is, if the test is positive, it means a true positive, means the antigen is really there. So this is one situation that occurs. Sometimes you have a test that is negative, but really the antigen is there. And in this case, this is called a false negative. When you are calculating the sensitivity, you have to take the number of true positives and divide it by the true positives plus the false negatives, because although it's a false negative, that antigen really was there. And this gives you the actual sensitivity that you should be expecting from that analyzer or from that test system. So this will let you know if this particular system that you're going to put in place is able to detect tiny amounts of the antigen being present. On the reverse is specificity. Specificity involves the negatives, the true negatives. So this is saying when the antigen is absent. Will I get a positive result or a negative result when the antigen is not there? Which means you're only looking for that antigen. How specific? You're looking for that one thing. If the test is positive and the antigen is not there, this is considered a false positive. If the test is negative and the antigen is not there, this is considered a true negative. Either way it goes, the antigen was not there in either of these cases. In order to determine what the specificity is, you take the true negatives, which is your true answer of what you should be looking for, and you divide it by the false positive plus true negatives, and that would give you a percentage. And that would tell you if the test is able to specify that particular organism or analyte that you're looking for. And of course, in the laboratory, we are so kind of I like to say we're spoiled. Most of the time, we like for our systems to have between 95 to 97% sensitivity and specificity, not one over the other. We want it to be able to detect tiny amounts of the antigen, and we want to be able to detect exactly the antigen that we're looking for and not pick up other things that we are not looking for. All right, so sensitivity and specificity, therefore, are inversely related. Test systems rely on balance, and we want high sensitivity, detect small amounts of antigen or antibody, and we want high specificity, detects only the desired epitope that we're looking for. All right, so antibodies that you can see in the laboratory, and again, this comes up in blood bank, this comes up in serology. Polyclonal antibodies, poly meaning multiple, contains a variety of binding sites. This affects the affinity and the avidity, meaning how strong it is to one and how strong it is to the group. Monoclonal antibodies, large amount of homogeneous binding sites, right? So it's highly specific and sensitive, and it's looking for one type of antibody. So this binds to latex, which is one version of how it's done, fluorescent dyes, which is another version of how it's done, enzyme conjugates, which is another version of how it's done, and each of those versions detects bacterial and viral antigens. Latex is the substrate, fluorescent dye is the substrate, or enzyme conjugate is the substrate. The substrate is the substance that is utilized to connect whatever we're testing with the antigen or the antibody. Within micro, these are some tests that we use often to detect our microorganisms. Precipitin tests, and this means you're going to have a line pretty much developed. Agglutination test, you're going to have clumping develop. And then immunofluorescence, you're going to have an antigen-antibody reaction in the presence of a fluorescent dye to give a color. And then enzyme immunoassay, you're going to have antigen-antibody reaction using some sort of enzyme as kind of like a binder. Within the precipitin test, here are some examples: double immunoelectrophoresis, counter immunoelectrophoresis, nephelometry. Agglutination, some examples: particle agglutination, latex agglutination, hemoagglutination, coagulation, and liposome enhanced agglutination. Immunofluorescence, and then enzyme immunoassay, solid phase immunoassay, and membrane bound solid phase immunoassay. Of course, this is going to be more important in chemistry. Precipitin test detects a soluble antigen in a solution. Double immunoselectrophoresis is called autolony. That's the classic test, so you might see that come up. Especially if the other thing about these tests, if you are only taking, say, microbiology, again, the exam has the same amount of questions. So whether you're taking a generalist exam or the microbiology exam, they're going to have the same amount of questions. So on a microbiology exam only, they're able to ask you more questions, and that's where things like this show up. So autolony is a classic test which uses agar gel. Wells are there, you have serum, and you have antibody added to the wells. And then you're going to look for an antigen antibody to move towards one another, and they meet in what's called a zone of equivalent. So when they meet, it's going to form the precipitin line, and that's what you're measuring. Okay, so here's the thing. It's not really practical, right? Why? One, because it doesn't pick up everything, and two, it's time consuming. And everything is about time nowadays. So then there's counter-immunoelectrophoresis. Counter-immunoelectrophoresis combines immunoelectrophoresis with a current. So we're going to speed this electrophoresis up, and we're going to have a zone of equivalence where the antigen meets the antibody and forms the band. So we're not going to wait for it to migrate on its own. We're going to give it a little bit of motivation. And then the other one is nephelometry. And this is where we're going to add light scattering. When the antigen reacts with the antibody and forms a precipitin, you're going to see like the light sources change. The amount of light that's there is going to change. All right, particle agglutination. So what you have here is two stages to agglutination. One, sensitization. The antigen combines with the antibody. And sensitization occurs in different methods. Sometimes you have to let the reaction sit for like 15 minutes. Sometimes you have to change the temperature. Sometimes you have to add a light source. It just depends on antigen antibody. A lattice forms between the antibody-coated particles. And then you have direct and indirect reactions. The direct reaction is the antigen is detected by binding antibodies to particles. Indirect is that the antibody by binding antigen to particles. So direct method is you looking for the antigen. Indirect is you looking for an antibody that was produced as a result of the reaction. Particle agglutination. Particle agglutination uses markers, the latex beads, red cells, fluorescent markers, enzyme conjugates. Those are just different markers that can be used. Immunochemical methods, latex agglutination, which is very popular, is a carrier molecule. Each particle can hold tons of antibody binding sites. And what happens is the antigen is in the specimen. It combines with the specific antibody on that molecule. So this molecule is a carrier for that antibody. This test is a rapid test, which is why it is used. And you can have direct specimen testing in bacteria isolates, which means you can do this test from the swab that you receive of the patient or from a culture sample. So you have multiple ways to use it, which is very helpful in the laboratory. All right, so direct agglutination. Direct, again, you're looking for the antigen. Bacteria antigens agglutinate in the presence of a host antibody. Examples of this: febrile agglutinase detect antibody to Salmonella and Brucella. Weedall test detects typhoid fever for Salmonella. Wild Felix test detects cross-reactive antibodies of Rickettsia. Indirect or passive agglutination. This is where an antigen is going to be extracted from an organism and attached to a carrier particle. And then the host antibody binds to the antigen in a positive reaction. Example: cytomegalovirus, varicella, mycoplasma, and syphilis hemagglutination. Here's an example of what direct agglutination looks like, just so you can kind of see it. You have a latex bead, you have the specific antibody and the specific antigen, and they're bound together. You add the specimen, and then you get a lattice formation, and this lattice formation is what is measured. Okay, coagulation. Antibody bound to a particle to improve visibility of the reaction. Labile Staph aureus is an example of a coagulation reaction where protein A binds the antigen through the Fc portion and detects soluble antigens in spinal fluid and serotyping of antigens. The Staphylococcus coagulation principle has been applied to the detection of soluble antigens in acute and chronic meningitis. So you're looking for coagulation. That coagulation tests the formation of a clotted substance. Immunochemical method, coagulation, and again, you learn a lot more about this in actually hematology, immunoserology. So this is just an example. So you have protein A, and the specific antibody and specific antigen are representative. So these green little dots here are protein A. In this case, Staph aureus has the protein A. So this is Staph aureus, represented by this ball here, right? This is Staph aureus with protein A. And then you have, notice the antigen is the purple little ball, and then the antibody is here with the epitopes are here. So anyway, you have a specific antibody that bounds by the Fc fragment. And again, you'll learn about that. So this binds by the Fc fragment and attaches to protein A. And then you have the Fab fragment of the antibody is available to bind to the antigen. So the Fab is on here on these little ends, binds to the antigen, which can be used to identify the bacteria in spinal fluid or from culture plates, right? So together you form a nice coagulation system, but this is how it is done. It's done through the presence of protein A on Staph aureus. Staph aureus binds with the specific antibody. That antibody binds with the specific antigen, and then you get this nice clumping that is measured. Next, we have ELISA, enzyme-linked immunosorbent assay. ELISA is used for like HIV. This is where antigen or antibody bound to the enzyme causes the catalytic reaction. It is measured spectrophotometrically in a spectrophotometer. It's very sensitive, and the specificity can be enhanced by using monoclonal antibodies. So a lot of the ELISA methods use monoclonal antibodies. Types, there's more than 3 types, but the main types are non-competitive or sandwich type, and then there's a competitive type, and then there's the solid phase immunosorbent assay. So this is where antibody or antigen can be fixed to a solid phase to detect whatever you're looking for. Kits are available for detection of Giardia, Cryptosporidium, Chlamydia, Neisseria, and then antibodies to Legionella, Mycoplasma. It's a lot of different things. Here's what happened in the ELISA detection for antibody. Again, specific antigens is little purple, but now you have an enzyme substrate. So the enzyme substrate is going to help the reaction out. Specific antibody here with your Fab sections and your FC sections, and then enzyme conjugated antibody is this represented here. What starts out happening first is the antigens attached to the solid phase beads, normally latex beads that go inside of a well, or it could be a wall of a microtear plate. And then you add the patient specimen, and the antibody binds to this antigen. Non-specific antibody does not bind and is washed away because there's a lot of washing in this process, right? So the specific antibody is what is left. Now you add the enzyme conjugate. And when you add the enzyme conjugate, the antibody is going to be there and it's going to bind to the antigen antibody complex if it is present. It may not be present if it was washed away. The excess enzyme conjugate is washed away. Because you added an enzyme conjugate, now that you add the enzyme substrate, the bound enzyme substrate indicates the hydrolytic breaking down of the water reaction, and then you get a colored end product. The intensity of the color is proportional to the amount of antibody in the specimen. So if there's more color, there's more antibody present. So you see you had to have an enzyme conjugate added in order for this enzyme substrate to actually be able to work. Next is immunofluorescence. This is where you use fluorochrome dyes to absorb energy in UV range and convert the energy into light. The most common dye is FITC, fluorescent isothiocyanate with rhodamine counterstain. And you can label antibodies or antigens, which is why it's good. It's sensitive, but it is labor intensive, so it is not used commonly. You just need to know the principle. You can just look at this and see that there's a lot of steps to it. You have to have the specimen containing the antigen fixed onto a slide starting. And then you add the fluorescent conjugate antibody, and then you incubate it. And then hopefully they make a reaction, right? All right, and then, so this results in fluorescence, which is observed microscopically, right? And then you have the specific antigen fixed onto a slide. Patient specimen is added. If the specific antibody, if present, the specific antibody is going to bind to this antigen. You wash it to remove unbound antibody. Now you add the fluorescent conjugate antibody. And then if fluorescence is observed microscopically, it's there. So first one was direct. This was direct. The second one was indirect. But it's extra steps. So it's a lot and it takes a lot of time. How long you have to incubate it takes a lot of time. And it's labor intensive. So it's not used as often. All right, direct immunofluorescence, specimens fixed to the slide, like I said, antigen complexes to antibody, unbound is washed away, and then the excited fluorochromes give off a light that you can detect the antigen. It is used for Bordetella or chlamydia. Indirect, detect the antigen or antibody. Antigen utilizes the SS antibody that binds with the antigen, and then the second antibody with a fluorescent label binds to it. With the antibody, the antigen fits on a slide, reacts with the antibody in the presence of the specimen. We wash away the unbound portion of it and add the fluorescent label that binds. And then you can dilute this for quantitative results. Immunoserological techniques. So, of course, this is serology. You get a lot more of this later in that course if you haven't taken it. Measure humoral immunity. So, mainly, primary immune response is IgM. That's the first immune response. The anamnestic immune response is IgG. That's the second time around. So, you get large amounts of immunoglobulin produced the second time around. You can do an antibody titer here, and some of you who've had blood bank know about this. Measure the antibody that's present, and the reciprocal of the highest serum dilution is what in which the antibody is detected is determined as the titer. All right, immunoserological applications, precipitin tests. So for syphilis, VDRL flocculation or RPR both detect Reagan, and it is an immunoserological test. You can have direct whole particle agglutination. You can have counter immunoelectrophoresis, ELISA, and membrane-bound ELISA, which is very sensitive. That's it. Western blot is the confirmation test for HIV, which is an immunoserological application. And you'll learn a whole lot more about that if you have not taken serology or chemistry. On to the next course.